Okay, let's get started. Good morning, everybody. So if you don't know me, my name is Rajiv Gupta. I'm a principal scientist. I just joined EPSAT recently. So it's my great pleasure to introduce Professor Guri Johal, who came from Purdue, USA. Guri has been a long time friend and excellent scientist. Guri did his MS in PAU. That's where the link is. I am also from PAU. So after MS, he moved to Simon Fraser University in Canada, Vancouver. So actually, he told me he was with David Burlington, who were DG. So they overlapped there as a graduate students. So during PhD, then after he moved to postdoc with Steve Briggs, a big name in plant biology, and did a postdoc with him in Pioneer. So then he took position in Columbia, University of Missouri, Columbia, for 5-6 years, and then moved to Pioneer again for a couple of years as a senior scientist. And obviously, from his work, you can see he's a kind of real scientist, and he didn't like the company and went back to university again. So moved back to Purdue in, I think, early 2000, and now he's a professor there. So Guri has done excellent work. As you can see, I mean, people in plant biology, especially in plant pathology, so everybody knows him. So basically, during PhD, he did an excellent job on glyphosate, actually, you know, the most used herbicide in the world. So how this glyphosate makes these plants more kind of, prone to diseases. So that's excellent work. Then after that, in postdoc, he cloned the first ever gene, actually plant disease-resistant gene from plants in maize, actually at that time in late 80s, early 90s. And then he continued on, same kind of disease pathogen interaction, plant pathogen interaction, and very nice work on what we call disease lesion mimic mutants, and very nice study on that. Then he has done another very nice work on dwarfing genes, actually. Paracatic 2, they cloned from maize and sorghum, and I think it's even being used in sorghum, commercial varieties too. So basically, it's a different mechanism for dwarfing what we generally hear from this green evolution, that's GA insensitive mutants. So this one is a transporter for auxin. So long list. So another one, I think. So we were talking about, this. So a very nice work. I think it's going to be a good paper too. So he has cloned insect hypersensitive mutants from maize. So basically, so I have seen those plants myself because we had some collaboration in Pioneer. So if you put a mutant plant and a wild type plant and in a chamber and put some insects, so insects directly goes to the mutant plant, so he has very nice work done on that. It's not published yet. So one thing I must say about Guri is, I mean, in generally, people work on one thing and just publish something and move on to another one. But in his case, he has done very kind of deep understanding of mechanism, how these things work. For the HM1, for example, that resistant gene, I mean, he has figured the whole pathway, how it works, And same is true for this BR2, Burgatic 2, CRW1. And so I think, so in plant community, we think about, we dream of 1 or 2 paper in big journals. So he has published many actually in Cell, Science, PNAS, several papers. And most recently, I think he got an award from Purdue University as well as a university scholar faculty from university. That's a most recent one. And so let's welcome Guri. And one thing, I think he will be here tomorrow. If someone want to meet him one-to-one, just let me know. Thank you.
All righty. Well, yeah. Thanks a lot, Rajiv, for your kind introduction. And hopefully what I talk about is kind of in the same line as well. When I was in school, we used to hear this proverb or phrase, necessity is the mother of invention. And actually that exactly applies to what we needed to do and what we have done. And that's what I'm going to talk about today. Okay, so the focus of what I'm going to talk about today is how to enhance or expand genetic variation. Much of it is done in corn, but I believe it can be extended to any plant species. And we can talk more about that when we have discussion. And genetic variation is very important because it's at the core of everything that we do in genetics, in genomics, and plant breeding. And a key source of genetic variation is, of course, mutations. Mutations that can happen naturally, spontaneously, and they do happen because of the low fidelity of DNA polymerases, okay? And especially under stress conditions. Sometimes we don't appreciate that, but if we have enough, I guess, controls and populations to look at that, stress does impact that. Okay, but you can also induce mutations intentionally or at will, and which are called induced mutations, of course. And to give you a little brief history of that, two people who were the pioneers are Hermann Muller and Louis Stadler. He worked on Drosophila, he worked on barley, and they both showed that X-rays induce mutations way back in 1927, 28. But there was one big difference. Muller was working on Drosophila, which has a 10 days generation time. And Louis Stadler was working on barley, which basically you can raise only one crop per year, right? 10 days versus 365 days. That's a big difference. So although I have learned from other people, he used to be at the University of Missouri where I went after I finished my postdoc. And at the same position, when Stadler, he actually died, and his position went to his student, Jerry Newfer. And when Jerry Newfer retired, I got that position. So there's a connection there. Hermann Muller got the Nobel Prize because he was able to publish his findings a little bit earlier than Stadler. And Stadler, instead of getting a Nobel Prize, he got leukemia because he was using his machine that he kind of improvised and take it to the field and zap these barley plants, with X-rays. And not knowing at the time, he also zapped himself probably. And he died of leukemia in early 1950s. But he's still considered an icon in plant genetics, especially maize genetics, of course. And one of his key students is Jerry Newfer, and I'm going to talk more about him later. He did do one thing at that time as well. Barbara McClintock, when she left Cornell, it was Stadler who convinced her to go to the University of Missouri. And much of the work that she did on transposable elements was done at the University of Missouri under the protection that he provided. Although, it didn't last very long, the protection, and she eventually went back to New York. But much of the work that she did on transposable elements that led her to get her Nobel Prize, although by that time she should have gotten her second or third, it was all done under his leadership. So induced mutagenesis. I got this definition from this paper by Rieger et al. It can be described as the process of inducing any heritable change in the genetic material which is subsequently transmitted to daughter cells where it gives rise to a mutant cell or individual. Okay. And mutations can be induced by a number of means or agents. These can be any form of ionizing radiation, physical agents, it can be chemical agents, or genetic mutagens like transposable elements, which are intrinsic in many of these plant species, or you can have something like DNA coming from another organism like tDNA or so on and so forth. Okay. What I'm going to talk about today is chemical mutagens. And one that has become the method or the mutagen of choice is EMS, ethyl methane sulfonate. And the reason is it's one of the most efficient mutagens that causes point mutations, not chromosomal aberrations or deletions or rearrangements, but point mutations causes SNPs in genomic terminology. And the way it does it causes ethylation of guanine, one of the 4 bases in DNA, converting it to 6-ethylguanine. And when that happens, and during DNA replication, instead of guanine pairing with cytosine, which it normally does, it pairs with thymine. And that's when mutation happens. When thymine is placed instead of cytosine, opposite to guanine, then during the next round replication, it will be adenine. So its effect of EMS is transition from GC to AT. Single base pair change because of that. But it cannot go back from AT to GC. So it's all in one direction. That's something to keep in mind. Okay. And just to give you a brief introduction to how mutagenesis is done, I'm sure many of you probably have done it or have thought about doing it. So I'm just going to briefly describe how it is done in plants thus far. With the exception of maize, and I'll come back to that later, mutagenesis is done by treating the seed with the chemical mutagen. And to do so, the seed is soaked in a solution, certain concentration of solution, for a certain amount of time, and then washed thoroughly, and after that, the seed, which now considered as M1 seed, is planted in the field. The seed that you start with is M0, but once you treat with the mutagen, it becomes M1. Okay? And that's something you want to keep in mind as well. It's a little confusing sometimes. And I want to lay these things down because later on when we are going to talk about some sophisticated designs, these things will be important. Okay? And the seed collector from these M1 plants is called the M2 seed. Okay? So you give you an overview. Here is the Arabidopsis seed. Arabidopsis is one of the model species, and this is where much of the chemical mutagenesis has been done, and many of the foundations of chemical mutagenesis that led to tilling, many of you probably are doing tilling, were laid down in Arabidopsis. So seeds are treated with a dose of 0.3% or this concentration here for 8 to 12 hours of duration. And that M1 seed is then planted to generate these M1 plants, and the seed collected from these M1 plants is the M2 seed, which you then can plant individually if you like, although in Arabidopsis people just bulk them, okay? Bulk them in pools of tens or hundreds or more, okay? So what happens when the seed is treated with EMS? Have you ever thought about that? And to able to understand it fully, you need to look at the anatomy of the seed. Okay. So what are you really doing to the seed when you're treating with EMS, and what's happening to those cells which are in the embryo? Okay, so look at the anatomy of the seed. This is what you have. Let's focus on the embryo. There's a radicle, the cotyledons, and there's a plumule as well, the shoot meristem. And to look closely at that, this is what it looks like. That dome-shaped structure, these are leaf initials. This is the dome-shaped structure is the shoot apical meristem or SAM. Okay. So every cell, all the cells are there, and when you treat with EMS, all these cells will be hit. So the genome of every cell or the DNA of every cell will be infected. But then I have these two eyes here, and these cells are very important, and these are called well, they're not called, but I'm going to call them germ cells. Okay, and these germ cells, these two cells are the one which give rise to much of the body of the plant, but especially give rise to the reproductive structures of the plant in the inflorescence. So all other cells, this has already, their fate is determined, and their fate is to keep producing more meristematic cells. But the fate of these two cells is to produce the reproductive organs, flowers of the plant. And it has been shown empirically that there are two of these in Arabidopsis. Although there is variation from genotype to genotype, and also, if you can extend the development of the embryo in the seed somehow, then this number will change as well. Most lines have only two of these, but some lines may have one, other lines may have four, or maybe even eight as well. And that's very important to understand. And many other plant species will have this variation. They may have two, or maybe have four or eight as well. So you have to, depending on what you're working with, you first want to figure out how many germ cells are there in the embryo of your plant of interest. Okay. And that is all done empirically. And this is called genetically effective cell number as well. Okay. So these cells, when they're hit, the mutations caused by EMS in these cells are completely independent of the cells, independent of the mutations created in this cell. So there's no relationship. So any mutations here are different than the mutations here. Okay. So this is what happens. Let's say there are only two cells, and there's a mutation in one of the cells, and that impacts somehow the color of the leaf. It becomes a little brownish or albino. And those two cells, one half of the plant coming from this cell, the other half of the plant coming from this cell. Although it does not normally happen that way. There's no 50 to 50 contribution. It may be 70 to 30, or it may be, any kind of ratio. But just for the purpose of looking at it closely, so let's say there's 50-50 contribution. So this mutation, if you somehow, if you can select these sleeks and look at the ratio of the mutation among your wild types, you may have a 3:1 ratio, which is a typical inheritance of a recessive mutation. But if you select these sleeks, you may not see the mutation at all, that particular one. But if you pool all the seeds, which is what people typically do, the ratio, instead of being 3:1, it will be 7:1. If you see any ratio close to 7:1, it means there are two cells in the embryo which give rise to the inflorescence. And these are called two genetically effective cell numbers. So it's very important that you first figure that out empirically by looking at the inheritance of mutations, which you can see easily. Albinos, pales, necrotics, seedling little, whatever it could be. So what it means that the M1 plant is actually a chimera of mutations. So these mutations, if they're coming from this cell, are only on this part of the plant. Mutations coming from this cell, which are independent of these, different than those, are only in this part of the plant. So M1 plant is a chimera. Although you cannot see it, right? But sometimes you do if one of the mutations that can give rise to an albino sector in the M1 plant or pale sector. And that happens once in a while, and it's shown here. So there's a mutation coming from one side of the plant, and this is albino here. So cells are somehow flowers coming from this side of the inflorescence are albino. From this side, they're not. So they will segregate 3:1, no segregation here, overall 7:1. It's very, very important that you look at the biology of your embryo in terms of what the genetically effective cell number is, because that will guide what you want to do with your tilling populations later on. And these two guys, way back in 1987, they estimated that you need roughly 125,000 M1 plants to fully cover or saturate the genome for all possible EMS inducible base changes. And this, this number is fairly effective. You can, people have been using this technique to generate mutations in any gene of interest, but still this number is not very, encouraging. It'll be much better if we can somehow increase the number of mutations or decrease this number to, let's say, 25,000, fivefold. Okay. In fact, people have been using different kind of ideas and techniques to achieve that. And there's a recent paper from Luca Kamai's group at UC Davis, and they showed that if you use tetraploid lines of Arabidopsis, then you can use those lines with much higher doses of EMS and can cause mutations which are 4 to 5 times or 4 times higher than if you use a diploid. And of course, once you have tetraploid, then it takes time to diploidize it as well. But again, the conclusion is polyploid, it allows the production of smaller populations for doing tilling. And that's very, very important. When you're doing these kind of reverse genetic approaches, if your population is very large that you need to screen, chances are you're going to fail. But if you can decrease the size of the population as much as you can to look for your mutation of interest, the chances of success will go up tremendously as well. So all these people are trying to do that as well. That's all I'm going to talk about Arabidopsis. I'm going to move to maize, because maize is a unique plant. It's unique because we cannot do, or we don't do, I shouldn't say we cannot, but we don't do seed mutagenesis in corn. Okay. Why? Because the biology of corn is very different. It's an open pollinated species, and it is a monoecious plant. It produces male and female flowers at different parts of the plant. And the germ cells, or so-called germ cells, which give rise to tassel, are different in the embryo than cells which give rise to the ear, the female inflorescence. And it has been empirically estimated there are 2 to 4 cells already laid down in the embryo in the seed of maize which give rise to the tassel. And similarly, 2 to 4 cells already there which give rise to the ear. So when you hit with EMS, mutations caused in this cell are different from this cell. Their mutations are different than this one. Every genome in every cell gets different sets of mutations. So mutations coming from here are different than this. Mutations coming from here different than this, and are different from all of these. But because this give rise to tassel, this give rise to ear, when we self these plants, we don't get these mutations in homozygous condition. We only get them in heterozygous conditions in the M2 population. As opposed to in other plants, in the M2 we get recessive mutations in homozygous condition. Okay. So that's one problem. To get mutations in a recessive gene, recessive mutation in a homozygous condition, we have to go one generation further to M3 to get them in homozygous condition. And if you're looking at mutations, for every 100 mutations, 99 will be recessive, 1 will be dominant. So the ratio is that skewed. But because in corn, it's open pollinated, so we have to self-pollinate to get these in homozygous condition. Having an extra generation to go to, it adds tremendously to the workload, to expenses and everything. So people found that unacceptable. It's just not feasible to do that. So they looked for other ways to somehow get around this problem, and and this approach or the strategy of pollen mutagenesis was invented. Okay. Just to give you an idea, so it's a maize plant. The ear is on the side of the plant, tassel is on the top. The cells which give rise to this one are different than the cells which give rise to those. Female ovules coming from here, pollen coming from tassel, and you have, of course, pollination. But these, we don't get mutation in homozygous condition in them. We have to do one explanation. So he's the inventor of pollen mutagenesis in corn, because by doing pollen mutagenesis, I'm going to come to that, you can get mutation in homozygous condition in M2. You don't have to go to M3, one extra generation, to get them in homozygous condition. And he was a student of his graduate student. So when he died, he got the position at the University. And when he retired, then I got the position. Although I did not stay there for long enough, I went back to pioneer. That's very nice. So how do people do pollen mutagenesis? Okay, so it's shown here in this slide. So let's say B73 is our golden standard in corn. It's kind of equivalent to Columbia if you are an Arabidopsis aficionado. So, so let's say you want to generate mutation in B73, and you plant B73, and from some plants you collect the pollen and treat that pollen with EMS. And then the seed that you generate and then after treating with EMS, you put that pollen onto the silks of a sibling plant. Okay? And the M1 seed that you generate from here is heterozygous for all the mutations coming from here. Mutations are not coming from the female side, only from the male side. And when you self-pollinate these M1 plants, then you get this M2 seed, which will segregate for mutations which are homozygous for recessive mutations. Okay. So we don't need to go one extra generation to get them in homozygous condition if you do seed treatment. But here we can, get these mutations in homozygous condition just in 2 generations. Okay. So these M2 families are then planted. So in corn we keep them separate. We don't pool them. We don't bulk them, as opposed to in Arabidopsis. And then we plant them and screen them for recessive mutations of interest. We will plant 24 plants from each of these M2 families, or 40, whatever you need might be. So this is called a random mutagenesis approach. You don't have really any interest in any gene or any mutation of interest. You basically develop this material, and this is kind of similar to what people do in tilling as well. You develop these M2 families, keep them separate, and then till them using whatever technique you might want to use to till. But pollen mutagenesis also allows you to target mutations in genes of interest. And you can do it only in corn because you're using pollen mutagenesis. You cannot do seed mutagenesis and target mutations in genes of interest. And the way it is done is shown in the slide. Let's say you want to generate more mutations in this gene called BRCA2. BRCA2, it controls height. Okay. When this BRCA2 is defective, the plants will be short. Okay. So if you use these plants, mutant BRCA2 plants, which are homozygous for the mutation, and cross them with pollen that you have collected from B73 but treated with EMS, put them onto silks of this plant and develop this M1 generation, fairly big one, few thousand to 10,000. And this BRCA2 gene allele coming from this one being dominant over this recessive all the plants will be normal. They're all tall. However, if this Burkitic 2 coming from this one gets hit with EMS, it will become mutant, and if that particular pollen is able to fertilize the ovule here, and you get the kernel, and that kernel when you plant will be short. It will be rare. And the mutation frequency, and people have discovered it using this kind of approach, is the best you can do is you can get one mutation out of 1,000 that you look at. People try to improve this by various methods, but this is the best they can do. But oftentimes it will be much less than that, because pollen mutagenesis is not only science, but there's also art involved in that as well. And it's very finicky, and only this kind of frequency will only happen if you spend a lot of time and effort into that, and you're really good at that. Oftentimes the frequency will be much lower than that. But the advantage is you can get multiple mutations in your gene of interest. You can have an allelic series. Some mutations completely null, some relatively weak, and everything in the middle as well. So we can do seed mutagenesis in maize. And again, this frequency is good enough to do the kind of stuff we do, but if you want to do tilling, it's depressingly low. And that's why we don't have any tilling resource in corn, despite all the things we have done in corn, all kinds of populations we have generated in corn, all kinds of transposons we have identified in corn. We don't have a very good tilling resource in corn. And this is because this frequency is very low, and we need to generate tilling populations which are practically impossible to handle, to do the kind of stuff we want to do with the reverse genetic resource. So there's definitely a need to enhance this mutation efficiency. But the problem is pollen mutagenesis is very low, and seed mutagenesis in maize is not done because we have to go one extra generation. So are we stuck? Yes. If we don't step outside the box and think of new things. Okay. And that's where the necessity becomes the mother of invention. So we needed to do here something to generate at least resources for tilling population. But you may have noticed up until this time, whether mutagenesis is done by the seed mutagenesis method or pollen mutagenesis method, mutagenesis is done only once. But if you have breeders, you can do recurrent selection. So why can't we do recurrent mutagenesis? That was the question that popped in our mind, and we addressed that. And thus, can we do recurrent mutagenesis approach to enhance the frequency of mutation? And in corn, the way we did it is, again, we did one round of mutagenesis the way we normally do, right? Generated M1 progeny, but instead of planting it all randomly, we planted that progeny into these paired plots or rows. And so it has undergone one round of mutagenesis here, and we planted these here, and then we took pollen from here, treated that with EMS again, and put that pollen onto the silks of these plants. And we keep these separate because we want to keep our mutation in a heterozygous condition all the time. We don't want them to get homozygous yet. So we did that, and we planted like 100 different plots in these paired combinations, and by doing that, we got 50 lots of seed, which we call BC1M2. So it has undergone one round of mutagenesis here, second one here, then we subjected them to another round of mutagenesis. Again, you follow this design, take pollen from this one, mutagenize, treat with EMS again, and put onto these cells. And at the BC2 M3 level, you get 25 lots if you start with 100 here, And from each lot, and you can scale these up and down. I'm just giving you a number here. And 5,000 seeds from lot 1 and so on and so forth, and you get from 25 lots lot of seeds. It has undergone 1, 2, and 3 rounds of mutagenesis. So the mutation efficiency has gone up 2.5 to 3 times. And that's quite a bit more. You know, when you're looking at population, instead of looking at 10,000, now we can actually do with maybe 4,000 or so. But still, it's not. It has taken us 3 years to enhance efficiency by 2.5 or so or 3 times. Can we do any better than that? I'll come back to that later. And actually, in fact, we have a what's different here? So we kind of introduced different seed mutagenesis methods. You know, we kind of combined. We were initially only using pollen mutagenesis, but we thought, well, why can't we use seed mutagenesis? Because the number of generations is not an issue anymore. So we combined these two together, and eventually what we came down to, I think, is really revolutionary. Okay, and it's shown here. So we take B73 seed and we treat that with EMS. Remember, the cells which give rise to Tesla are different, give rise to ERA are different. So we achieve 2 rounds of mutagenesis right there by doing seed mutagenesis of the seed. And then we can collect pollen from this one and treat that with EMS, and that will be the 3rd round of mutagenesis just in one generation. 3 rounds of mutagenesis just in one generation. We can plant the material again in paired rows as well. But before we do that, we actually can treat them with EMS again, and EMS does 2 more rounds of mutagenesis, one on the male side and one on the female side. If we want, we can also do pollen mutagenesis as well. And that will be 6 rounds of mutagenesis in just 2 generations. Right? But we think that this may be, we don't really need this one. We can stick with this one and then cross these plants, and we don't then have to follow this paired-wise combinations either. We can just do sim mating and generate a lot of seed right in 2 populations, in 2 generations, and we get a lot of seed, which is basically loaded with mutations, and every mutation is held in heterozygous condition. So you may ask that, you know, by doing all this, maybe by this time the plants look miserable. No, they don't. They look just like the B73 plants with occasional dominant mutations here and there that we see. But at this stage, the density of mutations is very high in this material, but their vigor or their appearance is just like B73 because every mutation so far has been held in heterozygous condition. And this seed here actually is the library of mutations. It has, depending on the number, you know, mutation and numbers game, depending on the number that you generate here, this library of mutations you can use for whatever purpose you want to use for. You can share it with your friends, with the whole community, and you can also derive from this one F2 populations, which you can tell. And you can, because the mutation density is so high, now you can use next generation sequencing to sequence index your tilling population. You don't have to use the traditional tilling method, which has problems, but you can actually sequence it. And instead of having tilling populations of thousands of plants, thousands of families, you know, you can shrink it down to fairly smaller population, maybe 1,000 or less than 1,000 as well. And that's where we are at now. We are doing it. We're doing it in collaboration with the fellow. I'm going to talk about him later. So the mutation density has been increased tremendously in this material. And not only that, this seed, once it has been generated, and we can share with everyone, no, people don't have to do mutagenesis anymore. It has already been done. The seed is there. You simply give it to them. And they can use the seed for whatever purpose they want. So, initially we used that for BC2M3, but now it's BC1M2. It's a library of maize mutations in which mutation density is expected to be many times higher compared to the traditional approach. This seed is almost an immortal resource of mutations and can be dipped into at any time for any kind of mutation need. And every mutation in this resource is held in heterozygous. And of course, as I mentioned, you can take few seeds from each of these lots. And again, the lot size is up to you how you want to control it. You want to maybe have more of these horizontally, but less of these vertically. And you can generate utility population as well. Okay. But not only that, because you're not only restricted to mutagenesis with EMS. There are other mutagens as well. Remember, EMS can only convert G into A or C into G. It cannot go backwards. But there are chemicals, ENU, sodium azide, which actually convert A into G or T into C. Or some of them, they don't have any bias. So we can control, we can mutagenize here with EMS, and the next step we can probably use another chemical. That way actually we can enhance the spectrum of mutations we get. Just think, let's you're trying to generate a mutant which is resistant to a herbicide, and you want to introduce that gene in the active site of an enzyme. But if that active site is not GC rich, but relatively AT rich, EMS is not going to give you any mutations which will be of interest. You may need other kind of chemicals. So you have to consider those kind of things. So this design allows us to use more than one kind of mutagen to increase the spectrum of mutations that we might be able to generate. Okay, so some of the utilities, we can generate allelic series of mutations very, very easily, very, very effectively. And one thing I haven't told you about, when we look for mutations with this approach, we look for mutations at the level of a family. And people who have done QTL mapping, you know that you cannot use F2 populations very effectively, but you can use F3 populations very, very effectively because you're looking for changes in relative which are subtle. Same thing here as well. And so, and you can to infuse new genetic variation into elite lines and also fix their potential drawbacks. And that's where this approach is very valuable, very attractive to companies like Pioneer, to Monsanto. Sometimes they have very, very nice elite lines, but they have one little flaw. Because of that one little flaw, they have to throw it away. But this approach allows that flaw to be fixed quite effectively. We can generate and maintain dominant mutation because we keep mutation in heterozygous condition. And it also allows us to explore heterosis or mutations which may be over-dominant. There are some cases of that. I'm not going to go into those. And of course, generate new traits such as herbicide tolerance. Because with this approach, we can generate mutant material which is as vast as you want it to be. And then you can zap it with the herbicide of interest, with the dose of interest, and look for variants which may be tolerant to different degrees. And this is actually shown if you want to generate new alleles of a gene of interest, and BRCA2 is one of the dwarfing mutations. And we are really interested in that because it allows us to generate cone plants which are of optimal height, not too tall and not too short. And it's been shown that there's a height of the plant which is really optimal. And when you go beyond or below that, then the yield takes a dip. So we basically use this library of mutations. This is again done with the BC1 less with BC1M2. So all you do is take 1,000 plants from your source, from all of these lots. Let's say you have 50 lots, you take 20 seeds from each of these, and cross them with your mutant of interest, which is, so you take pollen in this case, and you can collect that pollen in a bag, and slowly you can, in 10 minutes, you can make 100 pollinations. And that's the beauty of corn. Yes, you have to self-pollinate it when you want to but making those kind of crosses and the size of the progeny that you get is tremendous. So I can make hundreds of these. I've done it. I can make 500 pollinations in a day, very quick, very easily. And each one of them will be successful. The successful rate in maize is 100%. And so these plants, some of them may have Gilbert-Kittig 2 mutation in heterozygous condition. And if they do, when you make this cross, they will segregate 1:1. And because you're looking at the level of the family, you may have to plant 20 plants here or more than that. Even subtle mutations in that mutation can be identified now. Subtle mutations in these genes or mutations in genes which have a subtle effect as well, the QTL, they can be identified for this technique as well. Actually, that's why we got into that, but I don't want to get here. So I don't want to get into these, but that's so you can develop your tilling population with this as well. Once you have generated the material, then you can take few seeds from each of the lots and self them, generate F2 populations from these, but you can also sequence index them as well, and it will be highly effective. That's something we're doing now. Okay. I already talked about that, but I just, before I close, I want to acknowledge Brian Delix, who is an associate professor now in horticulture, and he's an expert in bioinformatics, next generation sequencing, genetics, and genomics. And Rajdeep, he was here a few weeks ago, and he's a graduate student in my lab, and he has been able to do all of these rounds of mutagenesis. It looks like a lot of work, but again, when you do either one reaction or 10 reactions or 50 reactions, it can be easily done. So Rajdeep is the person who has done that. And I think I'll stop here and entertain any questions that you might have. Thanks. Yes, please. Yeah. So that's why we did what we did, right? The way when after you generate M1 population, instead of growing them randomly in the field, we are growing them in plots. And when you can do paired pollination of EMS mutagenesis. And the idea of that is to keep every mutation in heterozygous condition. And that's how we do that. And you can do that only in corn, of course. You cannot do that in other plant species because corn is cross-pollinated and other plants are self-pollinated. Also, eventually, if you keep on doing it for more and more generations, there will be some duplicacy. That's why we want to also keep the number of generations as few as possible. That's why when we combine seed mutagenesis and pollen mutagenesis, in just 2 generations, we can have up to 6 rounds of mutagenesis. So we can have mutation rate 4 to 5 times higher than is possible ordinarily. So in corn, just by keeping it, every mutation it's in heterozygous condition. There's no duplication. So let's go back.
So maybe another way of asking the question is, does each M1 individual contribute only to only one late generation individual?
Yeah, exactly. So that's where you need to really look at the biology of your system. In corn, so when you hit with EMS either through the seed or EMS, the M1 plants that you get, each M1 plant has a different set of mutations. And even if you self an M1 plant in corn, you're not making them homozygous. You have to go, you're consolidating the mutations coming from the male side and the female side. So you have to go one extra generation by selfing twice to get them in homozygous condition. But that's not the case with other plant species. That's the case with only with maize. And the design we talked about, we have looked at this in all possible directions. So by doing the way we are doing things, we are keeping our mutations in strictly heterozygous condition. It is a lot of work to do that because we are doing pairwise combinations. But yeah, I know it was initially kind of confusing to us as well, but that's what happens, yeah. So, all right. Yeah.
So, sorry, but you haven't actually answered the question.
Mm-hmm.
So if you have a mutation in the M1 in one individual, in how many M3 individuals in your scheme will that mutation occur in the heterozygous condition?
Okay, so this is the one we, so let's say you got one mutation coming from here, right? And let's say it makes all the way through here. Yes, if you, when you do the pollen, to do the, not in the ear much, but in the pollen, and it can happen in the ear as well, then more than one kernel may have the same mutation, right? But there are multiple mutations in there, especially coming from the pollen. Corn produces so much pollen, millions and billions of grains of pollen. So every mutation is amplified there. But when you use that for pollination, only about 200 to 500 are actually used in there. So there will be some duplication of your mutation in there. But in this, yeah, so in this seed, there may be, if you have 5,000 seed, a mutation of interest may be more represented more than once here. It's hard for us to tell exactly how many times more, but it may be only once as well. It's numbers game, yeah. But the lot here has different sets of mutations than the lot here, than the lot here. That's why when we dip into that, we don't want to generate our killing population only from this lot. We want to take some from here, some from here, some from here, some from here, to make sure that we have as many independent mutations as possible. Is that good? Thanks.
Excuse me.
Yeah.
So you are keeping the mutations in dominant conditions, heterozygous conditions. You have said that only 1% of the mutations are in dominant mutations.
Are dominant.
So how do you identify the recessive mutations?
Yeah, so you will not see, if you go to this seed log, the only mutation you will see up until here are dominant mutations. You are not seeing recessive mutations. To see those, you have to self. Take 50 kernels from here, 50 kernels from there, 50 kernels from there, and if you have 50 lots, altogether these are 2,500. You self them. And that's what you actually use for generating your tilling populations. And so you can sequence index them, but you can also plan them as well and look for mutations of interest at that level. So for example, you want to look for herbicide tolerance or something like that. We are hoping that herbicide tolerance may behave as a partially dominant. There are some cases of that. Then we can generate a lot of material here and screen them at this level. We can generate millions and millions of seed here. This procedure can be scaled up and down. But if you have to develop F2 families, Yes, that's where the amount, that'll be a lot of work. So we cannot go beyond, if we're going to self these, we can self easily up to a few thousand, but going beyond that will be a lot of work. But we're hoping that for generating herbicide tolerance or other kinds of tolerance, heat tolerance, we probably, those will be partially dominant traits. Good question. Yes, please.
Question pertains to the delivery of this technology, this technique. With all this work, means you could find any inbred or line in maize which was of trait of interest to the corn breeders.
Mm-hmm.
Any such line was developed with this?
No, we haven't so far. But we're talking to Monsanto. So they have given us a couple of lines, or Pioneer. They have some problem with the line. We think we can fix them using this technique. But you can do, basically, there's potential, so you can do anything. So what kind of trait do you have in mind that you would like to generate by this, or you would like to get rid of by this?
I was just wondering, means corn people, they are working, so what type of variation they are not having in the germplasm available? So they have to go towards this this type of technology.
Yeah. So all these companies have beautiful elite lines. Their ability to yield is higher than anything else you can imagine. But they're not perfect, right? Especially when the conditions, the environmental conditions are changing. So that's where people are finding, and it happened in 2012 that the environment in the Midwest was very dry. It didn't rain for 3 months, and it was also very hot. There were temperatures about 40 degrees for 2.5 weeks at the time of flowering. And that's when people realized that elite germplasm is very, very susceptible to these kind of fluctuations in the environment. Much of the corn crop in the Midwest failed because of that. People had to plow down their fields. So these elite lines are wonderful, but the amount of genetic diversity in them is very limited now. And if you want them to adapt to new variations in the environment, you have to have diversity to be able to do that. This technique can infuse that kind of diversity in your elite lines. Yes, please.
In case of seed, we use LD50 to decide the concentrations and durations. In case of pollen, how do you do that? Is it by pollen germination test or what? To decide the concentration and duration.
Yeah, pollen mutagenesis. Yeah. So the people have empirically identified those doses. But it's a good idea, because there's a lot of difference between different genotypes, different immunolines. It's a good idea actually to take advantage of that information, but don't take it blindly, and do a test. Okay, so that information, that dose, definitely you can use that, but few other doses below it and above it, and do a test with your own genotype as well, and then pick up the right dose. Okay. Yeah, no, this is how we do that. But again, if you want to extend this idea to a self-pollinated species, it can be done as well. Again, people do mutagenesis only once and then generate M2 material. But if you want, you can go to take your M2 seed, few from each family. Let's say you have 10,000 or 1,000 M2 families, right? You take few seed from each family and mutagenize it again, and then develop M4 families. The mutation rate will be much higher there. If you're more ambitious, you can go to one more generation as well. So it's not only restricted to corn, but I think you can extend it to any kind of plant species. Of course, in other plant species, it's you cannot maintain your mutations all in heterozygous condition all the time. But that that's not it's not an absolute requirement either, whether your mutation is, as long as your seed is viable and it is performing well and you're generating more and more mutations, and then you're able to develop, your trilling population, not in M2, but maybe M4 or maybe M6 as well. Yes, please.
Yeah, I'm struggling with the problem with maize being cross-pollinating.
Mm-hmm.
Like you've got a B73 background,
Yeah.
and all of the seeds are B73,
Yeah.
and they've been mutagenized. And so if you have a field of maize,
Yeah.
and you allow them to cross-pollinate each other, i.e. you have a field of mutagenized maize, M1, and you let them cross-pollinate with each other.
Yeah.
That's kind of like self-pollinating Arabidopsis.
Well, it is, but again,
And it would
even if you
a lot less work.
even if you self-pollinate an M1 plant in corn, you don't get your mutations in homozygous condition. You only consolidate mutations coming from the pollen side and the ear in the M2 plant. That's why you have to go to one more generation to get them in homozygous condition in M3. And that was the reason why people invented pollen mutagenesis. Because those two cells, 2 to 4 cells, which give rise to tassel and ear, in the seed they're different. Mutations caused in the cells which give rise to tassel are different mutations. Mutations in the ear are different. Even if you self them, you're not making them homozygous. So you can self them if you want as well. But again, just for the convenience of things, and we want to keep everything separate and so on and so forth. And again, we are using this paired rows idea because you want to have enough pollen that you want to treat with EMS to produce the seed. Because if you take pollen from only from one plant, it may not be enough. That's the only reason why we are doing this way. But if you can do plant-to-plant mating, even following pollen mutagenesis, that would be fine as well. Yeah. Yes, please.
I think it's a very nice scheme for keeping heterozygous mutations and adding more in each generation. But you also lose them.
There's a dilution effect, definitely, every time
but it's just simply a segregation effect. So if you've got N heterozygous mutations and you keep the population size, in the next generation you've got a half N. So you're adding, if each time you're adding, you sort of asymptotically come towards the level of mutation you had in the original population.
Yeah, exactly. Yeah.
So does that sort of gives you a limit to the number of heterozygous mutations you can keep?
Exactly. So that's why we wanted to have only 2 generations instead of 3. There's a dilution effect because of segregation as well. So in if 2 generations you have 6 rounds of mutagenesis. But at the end of these 2 rounds, you have not increased your mutation rate by 6. You may have increased it only by 4. But that too is tremendously higher than only 1. Okay. So it's difference whether you have to look at 1,000 M2 families or only 250 M2 families. Yeah, no, I sorry, I see where you come from. But we can play some other tricks as well to increase the mutation efficiency. It has been shown by, when we treat pollen with EMS and then we pollinate the silks or the ears. The way the silks develop, the silks from the top of the ear, they are very small silks. And the silks coming from the bottom of the ear, they're very long silks. Pollen which is highly mutagenized, it gets outcompeted by relatively less mutant pollen if you're focusing on seed at the bottom. So by pre-selecting seeds from the top of the ear, we can enhance mutation density beyond 1 or 1,000. Actually we have done that, but still it's not by 4 factors of 5. But combining all these things, when you start thinking, you must step outside the box, and there are all sorts of possibilities come into mind and can be combined together. I'm not telling everything that you're doing. We filed a patent on that as well. But yeah, this is the idea is recurrent mutagenesis, which can be used not only in maize but in other plant species as well. Methane, yeah.
Okay, I think we are already over time. Let's thank Guri once again. And again, he's here tomorrow as well, if somebody want to meet him. Thank you.
Yeah, thanks a lot.